Hello everyone, thank you very much for watching my video. In this presentation I will talk about the work of Becker, who investigated the current research regarding the possibility of life on Mars. I will start this presentation by giving a short overview of the research topic. Then I will talk about the specific editorial by Becker. I'll then add my opinion about the research and then talk about the outlook on this research topic. In the 1960s, the first successful exploratory missions to Mars took place. The planet seemed to look a lot like the Moon, which was a big surprise at the time. The first few insights did not really support the possibility of life. The planet seemed to be a freeze-dried desert with an extremely thin atmosphere exposed to deadly ultraviolet rays and cosmic radiation. For life to have occurred on Mars, water would have been an important prerequisite. In 1972, the Mariner 9 spacecraft sent back several images from Mars's surface. There were several large channels visible on these pictures, which appeared to be clear evidence that large amounts of liquid water had flowed over the surface. In 1976, the two Viking spacecrafts landed on the surface of Mars and, with revolutionary instruments at the time, investigated the topsoil for organisms, without success. The Viking labeled release experiments did detect evidence for life on Mars. NASA and other scientists later disputed these results, though, as being false positives. So the question of life on Mars remained unanswered. Moving on to the research paper I will present, as already mentioned, I will present this editorial from Becker. What exactly is the purpose of this research? Becker analyzes several studies of one research team in particular, which investigate the possibility of life on Mars. As it is an editorial, I will also talk a little bit more about the references made in this paper. To answer the question if there currently is life on Mars, and if Mars in its state today is habitable, Becker considers various claims for current and past life in the context of what is currently known about Mars and its geology. In a wide diversity of settings, a wide range of alternation minerals have been observed on Mars. These include zeolites, opal, and smectite. I want to show you how one of those observations was done with this tool from NASA. So this tool allows you to view imagery from Mars, and now I can exactly show you where the samples I am talking about were taken. Can choose here the Curiosity landing site, then fly to this area where it landed. We can now zoom in a little, and we can see here the landing site where the Curiosity lander landed, and then it went over here at this spot where it took these samples. This is what the drilled holes looked like. The one on the top left was a test drill, and the content of the other hole on the right then got scooped up and then got further analyzed. For the analysis, the Curiosity rover used several instruments. Without going into too much detail, I want to show you what one of those instruments looks like. This is the so-called Chemistry and Mineralogy X-ray diffraction instrument. In operation, a collimated X-ray beam from the X-ray tube is directed through a transmission sample cell containing powdered material. An X-ray sensitive CCD imager is positioned on the opposite side of the sample from the source and directly detects X-rays diffracted or fluoresced by the sample. The CCD detector is operated in single photon counting mode and then exposed to the X-ray flux. The CCD can then be used to measure the charge generated by each photon and hence its energy. All of the X-rays detected by the CCD are summed up into a histogram of number of photons versus photon energy that constitutes an energy dispersive X-ray histogram of the sample. Here on the right, again, the XRD plot is obtained by summing diffracted photons from either of the characteristic lines of the X-ray source. On the left, the X-ray energy dispersive histogram is obtained by summing up all of the X-ray photons detected by the CCD. The sample then gets filled into the rover. The instrument is specifically located here on the rover. You can see here how the lid opens up and the sample, which we've seen before, gets filled in there such that the instrument can analyze it. Now going back to Becker, he states that these detected alteration minerals may support a bacterial community of chemotrophs which obtain nutrition and energy by the oxidation of organic or inorganic molecules. These chemotrophs and metal-eating bacteria acquire electrons from metals that become oxidized. As you probably know, there is abundant evidence of oxidation on Mars, such as its red color, the result of iron oxidation. Becker has also analyzed various claims of research teams which had apparently found extraordinary evidence for life on Mars. He specifically mentions Joseph and his teams, which have released various claims for life on Mars. Let us take a look at these claims. Becker goes on to explain that Joseph and his colleagues reviewed several reports and concluded that iron-rich Mars provides an habitable environment for Martian organisms, including fungi and lichens. They've also detailed the fact that the mineralogy of the Endeavour crater is similar to the habitat created by tube worms and identifies tubular structures on the surface, as you can see here on these pictures. Finally, Becker concludes, although they provide this extraordinary evidence, they do not provide proof. Additionally, Becker also talks about hydrated minerals and metals which also seem to be abundant on Mars. Among others, these metals and minerals include jarosite, magnetite, and calcium sulfates, as identified by the suite of instruments on the Mars rovers and on spectra obtained from orbit, and also from analysis of Martian meteorites like Miller-Range 03346 and Allen Hills 84001. The interesting thing about this is that these findings are analogous to terrestrial environments, which of course are infested with a variety of organisms. Therefore, Becker concludes that this could have implications for Mars. This has led to suggestion that bacteria, algae, and fungi may have precipitated or dwell within the analogous mineral and metal substances on Mars. This may also account for green colors that have been observed on Martian rock and sandstone. This hypothesis, again, is no proof though, and some of these Martian minerals on Earth also have a natural green color. Joseph has also reported veins of white substances like calcium carbonate and calcium oxalate, which on Earth are secreted by algae and lichens. Additionally, he has also presented what he believes to be photographic evidence of fossilized spherical organisms that are cemented in layers of calcium carbonate oxalate. This is what this looks like. Becker criticizes this by stating that NASA has also observed these cement concretions. They did not refer to the possibility of life though, and did not determine what the cement-like matrix consists of. He concludes that the hypothesis of Joseph is interesting, but it is only a hypothesis. Becker additionally states that there is some evidence for calcium carbonate on Mars from other sources, but there is no proof. A major thing one must consider when talking about possible life on Mars is also the fact that the present surface conditions on Mars are quite hostile and deadly to the forms of life as we know it. Important to mention here are the high ultraviolet rays and gamma radiation. However, this does not necessarily need to be a hindrance to life. Joseph and his team cite at this point numerous studies that document fungi growing in highly radioactive environments on Earth too, also mentioning the Chernobyl nuclear facility. Becker counters this though with the fact that Earth has an ozone layer which filters out gamma rays. The team of Joseph also cites numerous studies demonstrating that various organisms can survive over a year outside the International Space Station. One of these studies, though, also mentions that their samples of lichens did experience DNA damage when exposed to Mars-like environmental conditions, with ultraviolet radiation combined with space vacuum causing the most damage. Becker adds to this that it is true that these exposed samples regain photosynthetic capability and can even reproduce again, but the long-term effect on future generations is unknown. Will they mutate and develop life-neutralizing disabilities? Will they still be able to reproduce in the future? Joseph and his team used these insights from radiation experiments to underline their proof and photographs on the surface of Mars, stating that these captured organisms, which they identified, just adapted themselves to these environments. Becker again asks for proof for these claims. A simulated experiment is just not enough. Becker emphasizes that to understand some critical aspects of how life on Earth or Mars evolved, it is crucial to understand terrestrial biogeochemical cycles, particularly related to the mystery of methane on Mars. But let's first take a look at oxygen. Over hundreds of millions of years, oxygen on Earth was produced via photosynthesis, and between 2.4 and 2 billion years ago, after the Great Oxidation Event, the interaction between an oxygenated atmosphere and the surrounding hydrosphere, coupled with the creation of protective atmospheric ozone, led to a proliferation of life on Earth. Becker goes on and cites various sources which report that oxygen has been detected in the atmosphere of Mars, within soil samples and in meteorites. Additionally, there have been reports that the atmospheric oxygen shows seasonal variations, increasing by 30% in the spring and summer. On Earth, the primary source of oxygen is the photosynthetic activity of algae, cyanobacteria, lichens, and plants. It must be said at this point, though, that the mean volume of Martian atmospheric oxygen has been estimated at 0.174%. Becker mentions the various explanations which discuss abiogenic scenarios that could explain the oxygen of Mars, including abiotic photosynthesis. Oxygen was not the only essential ingredient for the explosion of life on Earth. Methane is also an essential component of life. Becker mentions a recent study on marine methane seeps on Earth. It has shown that vast quantities of methane move through the shallow subsea floor and are consumed by carbonate-hosted microbial communities, including methanotrophs, and that the interaction of these organisms contributes to the process of global methane dynamics. Recent findings have detected methane on Mars, but its origin and nature are still controversial, since carbonate rocks are speculated to be present in Martian craters like the Gale crater, which also shows evidence of water in the ancient past. Further study of it and other promising areas on Mars will be crucial to understanding the origin of methane and even life on Mars. As already mentioned, water would have been or is an important prerequisite for life on Mars to occur. Becker cites several recent studies which claim to have observed large volumes of water and water ice in the southern and northern polar caps of Mars. These occurrences may also extend deep into surrounding territory. The researchers use different methods to investigate these occurrences. The possible identification of subsurface liquid water was one of the main goals behind the development of the Mars Advanced Radar for Subsurface and Ionosphere Sounding, short MARSIS. The sounder was launched in 2003 on board the Mars Express spacecraft and began to collect data in the summer of 2005. Because of the lack of clear evidence in the first few years, the water was thought to be located in greater depth than previously thought. Another research team, though, analyzed radar profiles from in between 2012 and 2015 and was able to find evidence for water trapped below the ice of the south polar layer deposits. They justified this by stating, strong radar echoes from the bottom of the Martian southern polar deposits are interpreted as being due to the presence of liquid water under 1.5 kilometer of ice. Becker states that these subsurface glaciers are melting because they are heated by unknown thermal anomalies. According to him, these findings support the hypothesis that deep beneath the Martian subsurface there may be thermal vents that have been colonized by archaea and bacteria, including sulfate-reducing organisms. On Earth, these organisms live without sunlight via radiolysis and chemolithotrophy. This was one of the reasons for the ExoMars mission, which should have launched this month, was designed to drill 2 meters below the surface of Mars in search of life. Sadly, as we all know, this launch did not happen, and just a few days ago, the mission was postponed to 2028. So, what kind of conclusion can we draw on this? Is there no life on Mars? Becker finished the paper with these words: "It is unknown with 100% certainty whether there is extinct or extant life on Mars, or whether Mars can support life above the surface." Joseph and his colleagues have provided the scientific community with a wealth of data. They may have even discovered life on Mars. However, although compelling, there is no proof. More evidence is required, including and especially the physical extraction and direct examination of these photographed specimens. There is evidence of atmospheric oxygen and methane, gases that on Earth are largely produced by biological activity. What the source for them is on Mars is unknown. It is highly probable various organisms would survive and may grow below the surface, perhaps in subglacial rivers and subsurface lakes of water ice or within thermal vents. Samples from Mars may provide the answers to these questions. I personally share this opinion. After reading a lot of research papers and applying my knowledge from past lectures, I would say that having this evidence from the past 60 years, I'm really looking forward to possible sample returns from Mars, as they could shed some light on these questions and even give us specific evidence for extraterrestrial life and also other future missions like the already mentioned ExoMars mission. In my opinion, many of Joseph's claims contradict the approach today's research should take. Raising a hypothesis is one thing, claiming to have extraordinary evidence but not providing enough proof is the other. The research of Joseph mainly consists of making claims concerning photographs, but is that enough? I think Bianciardi and his team describe this fairly well when they say that even though morphology is a valid criterion for biogenicity, it has not been proven as an undisputed indicator of biogenicity if it is considered alone. In his book, Malcolm Walter also talks about this point and even says that arguments and speculation can be fun around the dinner table, but to make progress we have to look. One could therefore even go so far as to say that Becker should not have considered the research of Joseph at all, because he gave him a platform and put him in the spotlight, but on the other hand he mostly criticizes and disproves his claims. I want to finish this presentation by quickly taking a look at the future in this research area. I think the following years will be pretty interesting regarding the topic life on Mars, because several missions will be happening then. Here, of course, the ExoMars mission, which will most probably launch in 2028, and one of the biggest steps, of course, would be a Mars sample return to Earth. This is, according to NASA, only expected to happen in 2033. This is what NASA imagines the sample return could look like. You have this sample return lander, which would receive collected samples by the Perseverance rover and shoot them up to a spacecraft in the orbit of Mars, and then bring them back to Earth. I thank you so much for listening to my presentation. If you have any questions, please let me know. I'm happy to try and answer them as good as I can. In the following slide, you can see my references. If you're further interested in life on Mars, I recommend you go and check them out. Thanks again and see you around.